Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I'm delighted to introduce a very special guest on the show. We have with us today Dr. Kirk Brown, who is the Vice President of Research at Alnylam Pharmaceuticals. Alnylam is focused on developing RNAi medicines to transform the way diseases are treated. In addition to driving RNAi platform innovations, Dr. Brown leads a team of CNS target biologists at Alnylam and serves as the research lead for ALN-APP, the first clinical CNS RNAi program. So to learn more about Alnylam's approach to RNAi or RNA interference medicines, let's turn to Dr. Brown now. So thank you so much for being on the show today.
Thank you very much for having me and for inviting me to share some of Alnylam's experience with RNA interference for you.
I'm looking forward to it. So just to begin with, I think I want to take a step back and ask you more about what exactly RNA interference or RNAi is and how it's differentiated from other forms of genetic medicines.
Yeah, great question. Thank you. Yeah, so at a high level, RNAi is a conserved natural biological process in all of our cells. It's a mechanism that's triggered by small double-stranded RNAs or short interfering RNAs as they're known. And upon entry into the cell, the siRNA interacts with a set of proteins known as the RNA-induced silencing complex, or RISC. And once it engages this RISC, it then can seek out complementary messenger RNAs based on a sequence-specific interaction. Essentially, the sequence of that siRNA interacts with the sequence of the messenger RNA. This mechanism is catalytic, which means it can continue to bind, cut, and release that messenger RNA until all of that messenger RNA has been degraded, and that transcript is essentially effectively shut off or silenced, giving it that RNA interference or silencing effect. Essentially shutting off the protein that is driving the disease or the toxicity at the source, upstream of the protein production. Now, one way RNAi is differentiated from other forms of genetic medicine, take gene editing, for example. The activity of an RNAi therapeutic acts at the mRNA level, not the DNA level. And so the effect is tunable. We're able to see a dose-responsive effect with different doses of RNAi therapeutics, and it doesn't impart any permanent change in the DNA. If you stop treating with an RNAi therapeutic, the mRNA eventually recovers and the protein then would come back. So that's one area of differentiation I think is important to point out.
And as a therapeutic or in the therapeutic space or realm, can you tell us a bit more about the broad applicability of RNAi technology across different diseases? And I know that you're leading a number of different programs in addition to the CNS program at Alnylam. And this includes cardiovascular diseases or the cardiovascular space, which we know is very important because cardiovascular diseases represent the number one killer in the U.S. And so can you share some examples from some of Alnylam's pipeline, and we'll get into more of the clinical data and all of that later as well. But could you just give us a broad overview of the pipeline at the company?
Sure. Happy to. As you stated, I can start with cardiovascular disease because there's a number of things we're exploring there. As you said, cardiovascular disease is a leading cause of death across the world. We believe RNAi Therapeutics, as essentially a new proven class of medicine, has the potential to disrupt the way we approach treating things like hypertension and high cholesterol and other cardiovascular disease risk factors. One example, I think, is that patients are often treated with multiple medicines and multiple treatments to manage their cardiovascular disease, requiring folks to take a number of different treatments, a number of different pills, multiple times a day or daily, which they then have to adhere to over the course of their treatment period. And this can be sometimes problematic for folks to stick to. And another element I think that adds complexity to hypertension is that the treatments that are available now, the effectiveness of them tends to ebb and flow over the course of a 24-hour time period, right? So trying to normalize that treatment is an area where I think RNAi therapeutics can probably play an important role moving forward. And so one way we can overcome this challenge is we've developed and discovered an RNAi therapeutic, which is now marketed by Novartis, which has been approved to treat some patients with high cholesterol. And importantly, and distinction from the examples I was sharing a second ago, really with just 2 injections a year. So the treatment lasts 6 months at a time, essentially controlling an individual's cholesterol levels with 2 subcutaneous injections a year. This speaks to the mechanism of action of RNA interference, the sort of prolonged activity. And now the same molecule is being studied in a large trial to understand really if lowering cholesterol can also lower the risk of cardiovascular events upon treatment. We've also explored another investigational RNAi therapeutic for hypertension. This is in collaboration with Roche. And in early trials, we've seen that we've been able to reduce high blood pressure with this RNAi therapeutic. And it, similar to the high cholesterol lowering agent, we're looking to expand to do a large study to explore if lowering or controlling hypertension can also reduce heart attack, stroke, and cardiovascular death with the treatment of RNAi therapeutic. Again, this is very infrequent dosing, which helps control and manage that sort of ebb and flow that is often occurring with other hypertension treatments. And I think you were hinting at a more recent clinical trial readout, which I can touch on now because it relates to cardiovascular health. Very recently, we announced positive phase 3 data with vutrisiran. This is an investigational RNAi therapeutic for the development and treatment of ATTR amyloidosis with cardiomyopathy. This was extremely exciting for us because it showed statistical significance across all primary and secondary endpoints. It was really a 10 out of a 10 hit for us. Just a little bit on the specifics, we saw a 28 or 33% reduction in composite all-cause mortality in the overall population, as well as the monotherapy population. And we reduced all-cause mortality by 36 or 35%, depending on which patient population, overall or monotherapy. And, in addition, on the secondary endpoints, we saw clinically significant benefit in a 6-minute walk test, Kansas City cardiomyopathy questionnaire, and the New York Heart Association class. So essentially all measures of heart health we are exploring in the study and saw statistically significant improvements. And really, the relative risk reduction compared to placebo was 36 or 35% respectively, depending on the patient population, which was an absolutely incredible result from this study. And so we couldn't really be more excited about what we've seen with vutrisiran, and we're very excited for the potential to provide benefit to patients with or individuals with ATTR amyloidosis with cardiomyopathy, hopefully in the near future. So just covering that cardiology point there as much as I could.
That's indeed very exciting, and congratulations on the positive data there. Really, really great things to build on, and looking forward to that. And in terms of the therapeutic that you mentioned, vutrisiran, is that what it's?
Vutrisiran.
My apologies. And so, what does it target specifically? I believe it targets the ATTR.
It's called. Good question. Thank you. It's targeting transthyretin, TTR, which is the factor that actually aggregates and accumulates in the various target cell tissues, whether it's the heart for cardiomyopathy or the peripheral nerves and the peripheral neuropathy form of that indication.
Right. And so could this therapeutic be applied in other indications where there is a dysfunctional TTR or in other indications?
This one is very specific for TTR amyloidosis with cardiomyopathy and currently for polyneuropathy version.
Mm-hmm. And you also had the CNS realm of things as well. And so what are some of the developments in that space?
Yeah, I mean, this is an area that's very close to my heart. It's an area where I focus a lot of my attention. My team focuses a lot of their attention. So if you don't mind, I can tell you a little bit about the origin story of CNS. Absolutely. Here at Alnylam, which is an interesting one. And then I can talk a little bit more about where we're moving with that conjugate strategy. Some years back in 2018, we had just seen the approval of Onpattro for TTR polyneuropathy. And the company was essentially laser focused on additional developments in the liver. And so there was a very small fraction of us that at Alnylam we call like a 10% or 15% time project. Other groups might call it like skunk works, where there's sort of a group of cross-functional scientists interacting and brainstorming and trying to solve important pressing issues. And a group of us got together to create new designs that we thought might be amenable to CNS activity, which when it comes to delivery to the liver, we're using either a lipid nanoparticle or GalNAc siRNAs. And so this would be a different design strategy to enable uptake into the CNS. And so we pulled together a nice design, and we studied these new conjugate designs in rodents in 2018. And our expectation was maybe 10 or 15% knockdown that we might see in the spinal cord and the brain. And what we saw was actually remarkable. It was 70% knockdown, and it was essentially knockdown for the duration of the study. So then we were really excited, and we were then tasked to try to translate this from small rodents to non-human primates, which we were able to do on the first trial. When we then can move to non-human primates, then we know we are able to probably move this successfully to the clinic. And I think, your first question was on genetic medicine. I think one of the most powerful elements of genetic medicine is when you know the gene sequence, you can design an RNAi therapeutic very rapidly. And the time from technology innovation, and I can gladly say that the CNS designs were based on a whole foundation of research that had been ongoing at Alnylam to develop a very potent siRNA that are very stable in cells, that are very specific to the gene that we care about knocking down. And so applying that knowledge to these conjugates enabled us to move very quickly through the research to the early development and then into clinic. And I'm happy to say from our experience in preclinical efforts in CNS in 2018, we dosed our first patient with ALN-APP in April of 2022, which is our first and the first RNAi therapeutic for the CNS. And 1 year later, we saw clinical translation of target engagement in the CNS. And these are given through an intrathecal administration, so directly into the CSF. They make their way up into the past the spine and into the brain and into the deep brain. And what we're able to see is target engagement and target knockdown in patients with early onset Alzheimer's disease. And we're particularly energized by the speed with which we were able to make that transition from preclinical to clinical and clinical translation. And again, that speaks to the incredible power of RNAi therapeutics and also genetic medicine in general. And so along that time frame in 2019, we signed a collaboration deal with Regeneron to help enable and speed the process so we could develop new molecules for the CNS in partnership with Regeneron. And so now that pipeline is filling with molecules that are targeting MAP-tau, Huntington's disease, SOD1 for ALS, obviously APP for early onset AD, but also we've initiated a study in an indication known as cerebral amyloid angiopathy, which is currently in phase 2. And this is also an amyloid-based indication, but in this instance, the amyloid accumulates along the brain vasculature and causes brain bleeding events, microhemorrhages, stroke, and dementia in those patients. So we believe that the mechanism of action of, as I've said, shutting off that gene at the earliest time point prior to the protein being made is applicable across many different indications, whether it's TTR, whether it's APP for AD, or cerebral amyloid angiopathy. And so, again, the power of this technology is that we can very rapidly design a potent and stable and robustly silencing siRNA in a very rapid amount of time and move it quickly to clinic to potentially help patients across a variety of indications.
And speaking of translating these medicines to the clinic, how would you speak to a patient or even healthcare providers, physicians, about an RNAi therapeutic in terms of how, number one, it compares to standard medicines in terms of efficacy, safety, and outcomes, and the administration and some of the other things, because I think it's such a new class. Although we've had mRNA vaccines now for a couple of years, that was also met with a lot of questions. And so RNAi therapeutics drugs themselves are fairly new still. And so what is something that patients and healthcare providers alike need to know, or what would you
Yeah.
like them? Yeah.
Yeah, well, speaking in general terms, and I've touched on this a bit, but I think I would think patients and caregivers would need to think about this because they are quite different than today's medicines. You know, a small molecule that you might take daily or multiple times a day, or an antibody-based therapy. All of these are targeting oftentimes a toxic protein or a disease-driving protein after it has already been made, right? So it's just essentially constantly being produced, and you constantly have to treat it, which is why there's so much frequency in the treatments of these other types of approaches. Whereas with RNAi therapeutics, we're acting before the protein can be made at the mRNA level. So you have to take a step back. So the protein is actually unable to be produced with an RNAi therapeutic present. When it's cleaving that messenger RNA, the protein can't be made. And so we're effectively working upstream of today's medicines. And a couple other elements of RNAi therapeutics that are quite distinguishing is that it's just simply the duration of action, right? As I've said, the effect is quite long lasting. For our experience with in TTR, and a new molecule coming down TTR, we call TTRsc04. We expect to give it potentially once a year dosing to control the expression of TTR in patients. And essentially our thought here is that with these long-lasting effects, it will improve patient adherence, patient benefit outcomes. We expect it to, a single injection to be quite durable. And at Alnylam, we have a term we call clamped pharmacology, which is essentially a single injection robustly and rapidly reduces the target expression of the gene we care about. And you can see that essentially it's just a straight line where that target is reduced for the period of 3 or 6 months, or potentially even longer. And this allows that treatment effect without any of that back and forth or up and down. I think that's one of the main key distinguishing features between that and, say, small molecules or more frequently administered antibody-based approaches. Clamping the production of the target really until the next dose maintains that beneficial treatment effect. And I'm happy to say that the data that we've shared with ALN-APP and the CNS gives a very comparable profile to what we've already been observing with our liver. In the liver, we give our treatments generally once every 3 months, potentially once every 6 months. And we can see from a single intrathecal administration of ALN-APP, we've seen a very similar rapid and robust silencing of the biomarkers in the CSF. And we see that same clamp pharmacology for months also in the brain. So it really speaks to, again, the design features. These are very stable double-stranded RNAs. They're chemically modified, so they're protected from endonucleases. They engage the risk machinery, and they work over a very long period of time, and they do so very safely. These are very safe and tolerated molecules, whether it's sub-Q, whether it's IV, or so far in the intrathecal space as well.
And so what can you tell us about Alnylam's, I guess, experience in really transitioning pipeline assets to commercial medicines, and how are you incorporating those lessons and learnings into your ongoing R&D plans and programs?
Well, as I pointed out, we can move quite rapidly now, considering many areas of high unmet need. We have 5 approved products now, so we've seen get to commercial space at helping patients, one of which is partnered or marketed by Novartis. The rest are wholly owned by Alnylam. And we've learned a tremendous amount as we move from research to early development to clinic, how best to design studies to support those clinical trials, and how best to design to get the answers we need to provide potential benefit to patients. And so now the goal is to apply that knowledge to many, many other areas, in addition to high unmet need areas targeting the liver and in the CNS. I'm pretty excited about where things are going to be going here in the next few years.
And you also talked about, of course, a lot of your assets partnering with larger pharma companies like a Roche or a Novartis. And so what has that experience been like, and how, and I would say that you've maintained your name in this space at the same time. Usually you see smaller biotechs or mid-sized biotechs kind of being, for lack of a better word, bought up or gobbled up by
Sure.
the larger ones. But I would say this is a testament to Alnylam for, standing its ground and being a recognizable name despite having those partnerships and not being overcome by them. So could you speak to us a bit more about how you've navigated and maintained that?
Sure. Yes. I mean, I think a lot of it is shared interest from the different groups. I mean, with Roche and our work in hypertension, and then I can speak more to my closer interaction with Regeneron over the years. I was sort of foundational in those interactions with them, and it really it was a partnership built on the excitement that what we were seeing. We were seeing a single dose lasts a very long time in preclinical species, and Regeneron was pretty excited about what they were seeing there and the potential. And Regeneron's very well known for its interest in genetics and genetic medicine. And so it was really a perfect partnership in my mind, a perfect collaboration. And really, it's been fantastic. It's truly collaborative. We interact with them. We share resources, ideas. They're incredible at building mouse models and studying mouse models, and we're very efficient at creating very potent tools to explore ongoing targets that are in the collaboration, but also new targets that we're hoping to nominate and enter into the collaboration in the future. It really has helped speed the time with which we would normally be able to bring medicines forward. I mean, our pipeline, particularly at CNS, has filled very rapidly because of that collaboration. I can certainly say that if we were completely on our own on that, it would probably be a bit slower than it has been, just based on the amount of time and resources that we're able to apply to it. So by combining our forces, we've been able to move things forward very rapidly and move things into the clinic, with more to come in the next few years.
And then speaking about another key stakeholder, I guess, in drug development now is the patient voice and patients and caregivers, and really involving them in the drug development process. It's become more of not a trend, I would say, but there's more realization of the importance of that. And so how is Alnylam including that key piece and that key voice in the drug development process?
Yeah, we often put patients at the forefront and center of our focus. We interact and listen to various patient groups, and I'll say an approach that we've taken more recently, I think in the last 18 months or so, at least on the research side, to address the things we're hearing and the needs from patients, is this approach that we've put forth called 225. And this is a research and early development goal to expand and rapidly produce RNAi therapeutics across a variety of new tissues, as well as the ongoing current tissues. So 225 for research and early development at Alnylam means that we will bring forth 2 new CTAs for CNS, 5 new CTAs for the liver, and 2 new CTAs in emerging tissue areas. So expanding upon our technology outside of the liver, outside of the CNS, into new target tissue areas where there's high unmet need. This would include the muscle and also into adipose. And these are areas now that we're going to be addressed and able to address more patient needs because we're able to access more and more tissues and target new genes in these areas to address the needs of the patient. So they are often at the forefront of our focus when we're trying to move. And in total, that would be 9 clinical trials authorized within a 2-year period, which is a lofty stretch goal, but we're happily well on our way, I believe, to achieving that within the next 18 months.
Yeah, it's great to see the patients themselves helping drive a lot of the research and development, and really seeing their involvement and keeping them at the forefront. It's so important. And so having been in the space for a while now. Where do you see the field going, and how have you seen it evolve up until now? And where do you see RNAi therapeutics in the field heading in the next 5 to 10 years in terms of potential breakthroughs or shifts in the landscape that you're excited about and looking forward to?
Yeah, that's great. I think fantastic question. I think looking back on the greatest challenges with RNA interference has really focused on delivery. I mean, natural double-stranded RNAs or short double-stranded RNAs are not really attractive looking drugs, right? They degrade very rapidly in the plasma. They're not able to survive long in the cell. They also could be immunostimulatory. So natural RNAs are not really sharing a lot of drug-like properties to what our new RNAi therapeutics have now. And I think solving the delivery question was one of the first hurdles, right? So the first approach was using lipid nanoparticles to deliver a partially stabilized RNA to the liver. And that's what's used in designs for Onpattro right now. The next jump or leap in technology was sort of the conjugate design, a fully stabilized double-stranded RNA, which also has GalNAc conjugated to it, which then essentially laser focuses it to the hepatocytes, which once again improved the technology tremendously, allowed for less frequent dosing, more durable response and activity. So that was a big leap forward. So, steps in making the drug sub-Q administration versus IV, that's a big improvement. In the last few years, we've seen a big push into CNS, so different design features that enable compartmental dosing, as I said, to the intrathecal space. This is a big new change that should help enable the treatment of a broad variety of neurodegenerative, potentially neuromuscular disorders, ALS, PD, Huntington's, and many more. So I honestly believe it's really a question of delivery to solve, and that's where I do see things moving. Things are beginning to move. We're moving. Others are moving into areas such as the muscle, into adipose, into lung, cardiac, heart targeting, and essentially it becomes just a delivery hurdle because, as I said, RNAi machinery works, and it works very well in most cells. If you are able to jump over that hurdle of delivery, once that RNAi therapeutic enters the cell, it's going to engage the target and silence that message, whether it's the heart or the skeletal muscle or the adipose or what have you. So I believe moving things towards targeting a variety of new tissues is going to be sort of the next wave of innovation coming from the RNAi therapeutics field. Maybe another big leap, or at least providing some potential optionality for the CNS would be that would be blood-brain barrier delivery. So a sub-Q or IV administration of a brain-targeting therapeutic, RNAi therapeutic, to deliver across the blood-brain barrier into the brain, I think is likely also another wave of design innovation that's going to be coming. The hope would be to move to a more convenient method of delivery and potentially enable access to even more patients with a sub-Q or IV administration versus intrathecal. Even if that intrathecal is quite infrequent, as I said, 6 months or so of dosing right now is very encouraging. It's a great first round, but we're hoping to continue to improve upon the designs there. And I do think new improvements in delivery, whether it's blood-brain barrier or targeted delivery to certain tissues, is probably the next more exciting wave of innovation in this space, which I can't wait to be a part of.
And thank you so much for the work that you do in leading those innovations and looking, we can't wait to see what those are going to be and looking forward to those as well. Dr. Brown, thank you so much for being on the show today. We really appreciate your time and insights.
Thank you so much. I mean, I believe we're right at the cusp of what we would consider here as an RNAi revolution. I mean, we're at the start. We're now showing very infrequent dosing in patients, robust silencing clinical outcome benefits across a variety of different high unmet need indications. We're just really excited about what the future holds for RNAi therapeutics. So thank you very much for having me on today.
Thank you. That's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.